It is time to start writing your unknown report. It is time to start writing your unknown report. And one of the first things to do you've already completed, and that is organizing all of the data for all of your results into your descriptive chart so that you don't have to scroll back and forth through lab pages or in your electronic notebook. You have one document where all of the results are. And you've probably come to at least a tentative identification of your unknown. And if you haven't done so, then that's where you need to go to that exercise 9 in the LaBeouf and Pierce lab notebook and follow along in those charts until you find your unknown. Let's scroll till I get there to exercise 9. Oop, that's the appendices. Here we go. And that if you have, move me out of the way so I can get to the right exercise. Giving you time to organize your notes. There we go. Chapter 9. If you have a gram-negative bacillus, those are a member of the family Enterobacteriaceae. And there's a list of possibles that are beyond what's on our list. Our list is that data for the dichotomous key. Those data are the 12 possible unknowns for the class. And with the gram reaction, you split your possibilities exactly in half. With the gram negatives, initially it seems a little unfair because nothing else in that chart got you anywhere closer. There's still 6 left at the end of that dichotomous key. All 6 of the gram-negative bacillus are facultative anaerobes, and they end up in the same group. I designed that dichotomous key that way, however, because for the gram-negatives, then you come to this table on page 547 that tells you need your indole, methyl red, Voges-Proskauer, and citrate results. We did not do citrate because if you look at this, all of them could either be positive or negative. So no matter what your citrate result was, it would not help you. So you just need your indole, which is exercise 518 from the SIM test, your methyl red and Voges-Proskauer, which are exercise 5-3. Find out if you have all negatives, if you have a negative, a positive, a negative, negative and 2 positives, a positive, and a negative. Whatever your combination for your indole, methyl red, and Voges-Proskauer tells you which table to go to next. And once you get to your next table, then it tells you things like lactose fermentation. You would look at your descriptive chart. Depending on if you were positive or negative, you follow that direction. Let's say you were negative for lactose fermentation. Then it says arabinose. We didn't do that, so you have to skip it, but there is another test. We did do lipase. And if you had a negative result, it'd be Hafnia alveoli, and a positive is Serratia marcescens. Keep in mind that wherever you end up on this flowchart has to be an organism that is in that list of 12 possibilities. Hafnia alveoli is not on that list. So that would tell you that you probably have Serratia marcescens. So either your lipase test was wrong, and there's nothing you can do about that, you were given the results, or your lactose fermentation was wrong. So you could say, well, if my lactose fermentation was correct, then I end up down here with Serratia marcescens. So that's one possibility for my unknown. But what if instead my lactose fermentation results were wonky, and maybe my unknown was actually positive for lactose fermentation? Then you would check your motility and ornithine decarboxylase. And if you were negative for both of those, then your possibility is Klebsiella pneumoniae. If you're positive for both of those, then you have to go to the lysine decarboxylase, and you should end up here at Enterobacter aerogenes because Enterobacter cloacae is not on our list. So if everything works out really well and they all work on one side, that you do the flow chart, you can be fairly confident that is your unknown. However, if you aren't certain, you'll end up with two possibilities and write both of those down because they are equally possible. There would be other tables within the gram negatives, but they work the same way. If you have a gram positive coccus, then your options are for either catalase positive or catalase negative. So if your gram positive coccus was positive for the catalase test, the one that bubbles, exercise 5-4, then you look for, it says the glucose oxidation fermentation and the bacitracin. We did not do bacitracin, and we didn't do the glucose oxidation fermentation test, but we did do glucose fermentation tests. And so this side, oxidation positive, means obligate aerobe, and none of ours are that. So if you have a gram positive, catalase positive coccus, you are going to go on the side where it is a facultative anaerobe. And here our options are Staph aureus, Staph epidermidis, and Staph saprophyticus. Aureus and epidermidis are on our list. So then the next important test is the coagulase test. If your organism was positive for coagulase, exercise 523, and it clumped the rabbit plasma, then you have Staph aureus. However, if it was negative for coagulase test, then we go to the next one. We did not do novobiocin, but we did do nitrate reduced to nitrite. And if your organism was negative for coagulase and positive for nitrate reduced to nitrite, then that confirms it as Staph epidermidis. That takes care of our gram negatives and our gram positive cocci that are catalase positive. The next one is the gram positive cocci that are catalase negative. The next thing you need is your hemolysis reaction. If it had beta hemolysis, complete hemolysis, you're on this side. And if it had either alpha, partial hemolysis, or gamma, no hemolysis, you're on that side. We did not do the PYR test, but what we can see is that if that test were negative, Streptococcus agalactiae and Streptococcus equisimilis are not on our list of possibilities. So you would have to be on this side, and these two are on our list of possibilities. We did do mannitol fermentation. You would need to look at your phenol red broth mannitol fermentation because the mannitol salt plate has high salt and killed these. But we do have the phenol red broth with mannitol, and if the fermentation of mannitol is negative, that leads us to Streptococcus pyogenes, and if the mannitol fermentation was positive, that leads us to Enterococcus faecalis. If our hemolysis was alpha or gamma, we have on this side only one possibility from our list of 12 unknowns, and that is Enterococcus faecalis. So we would look at the Vogts-Proskauer test. It should be positive because there's our Enterococcus faecalis. Our mannitol fermentation is once again positive, and our arginine decarboxylase positive, starch negative, would get us to E. faecalis. If you had alpha or gamma hemolysis and a negative Vogts-Proskauer, you're going to end up somewhere that doesn't work. So if that happens to you, let me know, and I can help you sort through your data to see which time did your unknown behave weird and help you get on track. And then lastly, we have the gram-positive bacillus. There we are. The catalase test, if it bubbled with hydrogen peroxide, exercise 5-4, should be positive because anything with a negative catalase result that is a gram-positive rod are not on our list. So you should be catalase positive. The next test is motility. The ones that are negative for motility are not on our list, so your motility should be positive. That brings us down to the mannitol fermentation. Again, you need to look at the phenol red broth for the mannitol fermentation because that is not selective. Everything grows in it, and then we can truly determine if it ferments mannitol. And if it's positive for mannitol fermentation, we look at the starch hydrolysis. A negative result is something not on our list, so you should be positive. And then we should be positive for the Vokes-Proskauer and negative for phenylalanine deaminase. You should end up at Bacillus subtilis. If you don't, and you end up over here, let me know, and I'll help you figure out your results. If we go back to our mannitol fermentation and it's negative in phenol red broth, then we need our casein hydrolysis. We did not do citrate, so our casein hydrolysis, and we should be positive for that, and that will lead us to the Bacillus cereus. That's how you have your tentative identification. Then you have to confirm that with a document called the Burgess Manual. In our face-to-face class, we have hardcover books, one for each table, and we get to look through and discover the tables that give us the results that Burgess Manual does. And that is a book where bacteriologists from all over the world do intensive biochemical studies to determine what are the typical reactions for that species. And there are tables in that book. Unfortunately, despite many years of me asking for an online version of that for the college, we do not have one. And you cannot get to the library to check it out. However, Julie and I have dug through the Burgess manuals and created a table. And that I have provided a link in the lab archives, and it looks like this, where we have the alphabetized list of the 12 possible unknowns. I have the gram stain with the gram reaction first and the cellular morphology second. And then across the top are the names of all of the tests that we have done on our unknowns for this semester. I do have the exercise number, so that helps you find it. And I have the results listed with symbols. Those symbols come from the lab manual. Each exercise in the lab manual has a table, and in that table it indicates what should the symbol be for the results and what is the interpretation of those results. So let's just look at the first one. The gram stain for Bacillus cereus is gram positive. Its cellular morphology is a bacillus. Its growth on PEA was good. Its growth on MSA, the mannitol salt, was good, and it was red. The growth on MAC was poor. The growth on ENB was poor. If you don't remember what the poor and good mean, in your lab book, every single exercise does have a table that lists the symbol and the interpretation. So you should have those symbols and those results in your lab reports and on your descriptive chart. And for the most part, people did a great job with results. Sometimes your interpretations were a little off, but your results were good. And then you could come in here and say, well, I thought I had Bacillus cereus. And you're going to create a table where you have the list of all the tests we've done, the list of the results for your unknown, and then this table with the list of the results for what should Bacillus cereus actually do. And the table you will create using this data should look like this. And I will have this table in the lab archives along with a link to that Google Doc for you to access all your results and with the citation that you will need. Because I did not come up with all of these results off the top of my head. I had to look them up in Burgess Manual. So I need to provide the citation for where I found those results, and then you need to provide the citation in your paper. So your comparison table, and I provide this document, you will not have to recreate it, lists all of the tests in the first column. They are in the order they are in because that's how we found them in Berge's manual. Then you list your unknown results, all here. And I have two columns, just in case you did get stuck. You had weird results for a test, and you're either this organism or that organism, then you need to put them both in here. If you had very reliable results, and when you went through the flow chart, you only went one direction, all of your results matched, you're only going to need to fill in one of the Berge's results here. And at the bottom, how many total matches? How well does the organism's Berge's manual reactions actually match up with what your unknown behaved? This is not the actual correct citation, so I will have to fix that so that you have the correct citation. But this is your comparison table. You are going to be required to have this as one of the pages of your unknown report. That shows me you went through the flowcharts in the lab manual, and then you took that information and compared it to what are known results for your hypothesized identification, and that is what confirms your unknown's identification. So in the lab archives for week 10, I'm going to be posting this video in the lab manual, and then access the Google Doc. So when you click on that Google Doc of all the results for all the unknowns pops up. Hopefully it hasn't logged me out. Then I have said to completely fill in all your results into your comparison. You need to type in your answers. And then I have the blank document as a PDF and as a Word document. And then once again, the instructions for how to upload your file. Please don't take a picture of it. Upload the file itself, because then I can open it on my computer and type in comments directly on that for you. And then that is your comparison table. The second lab this week will be watching a microbes and disease video that I have to find the embed code for, but I will. And then there will be some questions associated with that.